Many flies are needed to produce medicinal quality larvae. The fly species used is the common greenbottle fly, Lucilia sericata. The number of fly cages depends on the numbers of flies needed to lay the required quantity of eggs, which develop into the larvae used for the product. The fly cages are covered with ladies' tights to stop the flies from escaping and to allow the easy transfer of materials into and out of the cages via the cut-tight legs. The flies prefer a moist, dark and quiet place to lay their eggs, so the egg collection devices consist of a dark plastic container prepared with a special medium, whose odour attracts the female flies to lay their eggs. Small plastic strainers are placed into the medium inside the container to facilitate the egg collection. The containers are placed into the fly cages and left for a few hours for egg laying to take place. After this time, the containers are removed and the eggs collected, weighed and used in the production process. The collected eggs are used for two purposes. The largest number are used to produce the medicinal quality larvae, which is the active ingredient for the larval therapy products. A small amount of the collected eggs are used for stock breeding. These eggs are placed on a special medium in plastic boxes used to rear the larvae. After collection, the eggs are processed to produce the active ingredient of the medicinal product. The fly eggs are washed and separated in a controlled environment. A small fine mesh sieve is used to aid in separation of the eggs to ensure that individual eggs are available that have their whole surface accessible for the disinfectants. After the initial disinfection, the eggs are transferred into the clean room. The next steps are performed in a Class A clean room cabinet. The first disinfectant solution is washed off the eggs before they are transferred into a second disinfectant solution. After the final washing step, the eggs are resuspended in a sterile saline solution and transferred onto specially prepared agar plates using a sterile pipette. A sterility test of the disinfected fly eggs is performed for each sublot prepared. The fly eggs hatch on the agar plates and develop into larvae in the required growth stage. Before use, each sublot is tested again to confirm the sterility of the larvae. Only sterile sublots are released as the active ingredient for production of the final product. The filling and dosing of the final medicinal products is also performed in a Class A cleanroom cabinet. The entire process is closely monitored for microbiological and particle contamination to ensure a clean environment throughout this critical production step. The final products are transferred into sterile plastic tubes, the primary container, and labeled with the batch, expiry, and product information. All raw materials used for the production of the medicinal product are sterile and approved for use by the quality department. The final products are transferred from the clean room into the dispatch area, where the tubes are sealed, labeled, and packed into transport boxes, ready for dispatch to customers.